Okay, good morning, students. In the earlier classes, we have discussed the alpha beta spectrums and how the decay happens and Fermi Curie plots and all the interactions of gamma radiations and properties of nucleus. And also, we have discussed the deuteron problems, that is two-body problem, and we have discussed some problem models on the nucleus that are shell model and liquid drop model, and the semi-empirical mass formulas. And also, today we are going to discuss about some techniques, and also there is a I have left one of the topic, that is meson theory, meson theory of nuclear forces. So let us discuss this meson theory of nuclear forces, and we will go to the material characterization techniques. Okay, how the characterization will happen. So I will share the screen. So this is the meson theory of nuclear forces. How this theory helps to understand the nuclear forces. The range and power of nuclear forces were topic discussed in the earlier sections, and here directly we will go into the theory. That the well-known Yukawa theory of exchange of forces of mesons by nucleons offers a solution. These meson exchanges produce exchange forces. Let's examine the fundamental aspects of Yukawa's concept. There is a concept of Yukawa which helps to understand the nuclear forces. Let us see that those fundamental aspects. Nuclear forces originates from exchange of particle with non-zero rest mass called meson. So nuclear forces, from where these nuclear forces originate? From the exchange of a particle, from the exchange of a particle with a non-zero rest mass. Okay, which has having a non-zero rest mass. From that it originates. Actually, the nuclear forces, those are called mesons. Okay, each nucleon is surrounded by a meson field. As a result, it continues emits and absorbs mesons. This exchanger of meson give rise to nuclear force, nuclear forces, and exchange of meson leads to constant further of momentum from one nucleon to other, and hence the force is exerted between them. So for every nucleon, there will be meson surrounded to those nucleons. Due to those nucleons, there will be exchange happens. Due to those exchanges, the nuclear forces will be evolve. Okay, the emission of meson ought to reduce the mass of nucleus. However, this is not observed. Thus, exchange of meson must take place in such a short time that the uncertainty in the energy is in consistence with the Heisenberg uncertainty principle. So, this makes the detection of exchange impossible, and so on. So the meson in the exchange process is referred to as virtual mesons. So those detection of these, the emission of mesons reduced mass of the nucleon, right? So this is not observed. The mass of the nucleon will be not emission of meson will not observed. The exchange of meson takes place in a very short time. Okay, it is done in a very short time. So the detection of meson exchange is very impossible. Okay, so the meson exchange, we will consider it as a virtual mesons. Okay, the meson responsible for strong nuclear forces are pi mesons. Okay, the force between proton neutron is due to pi plus and pi minus. Okay, so what is the responsible for these strong nuclear forces means pi mesons are the most responsible particles, which helps to bind that nuclear forces by to have that nucleus as strong, containing strong binding energy, because these pi mesons are responsible for these nuclear forces. The force between proton and neutron is pi plus or pi minus. The force between this proton and neutron is due to the pi plus and pi minus. When proton is disintegrated, when proton is changed to neutron, it will release pi plus. This is pi plus now. So if neutron is changed to proton, it will release pi minus. So there will be exchange of these ones. In the similar manner, proton, when it is changed to proton, excited to, so it will release pi naught, and in the same manner for the neutron also. So these are the actually meson theory of nuclear forces, Yukawa theory, we will say. And there are some estimation of mass for the uncertainty principle we'll discuss. So if a nucleus emits one meson, it loses an amount of energy m pi c square, isn't it? So when the virtual meson of rest mass is m pi, which emits nucleon, it will lose some energy. The energy is m pi c square. That is delta is equals to m pi c square. Proton changes to proton, and it will release a pi naught. Now the proton mass is above, transition must decrease, which is not observed in the experiment. This violation of conservation of energy is understood if the meson is observed again in a short time, delta t, such that delta E dot delta t nearly equals to h cross. So the complete reaction is given as, so the complete reaction will be given as like this. P gives rise to P plus pi naught, and it is given to P, that is proton. The emission and absorption of a particle with rest mass m pi c square is consistent with the uncertainty principle. If the particle encounters some same and another nucleon in time delta t, given by delta t nearly equals to h cross by m pi c square, even if we assume meson travels at the maximum possible speed, that is the speed of light, the maximum distance it can cover in this time is c dot delta t. So what is the maximum distance it can cover? That is velocity into time. If it moves, if the meson moves with a velocity of light also, it will, the velocity of light c into that time, we will get the maximum distance, how much it ranges, it goes far. Okay, that is r is equals to c into delta t, speed into time, h cross by m pi c. Okay, this is delta t is m pi c square, and here we have c, so c will get cancelled, we will get h cross by m pi c. Okay, m pi is equals to how much? H cross by rc, you can write. So from this equation, we can write m pi is equals to h cross by rc. These are the all constant values. So we have taken the values and we got the value as like this. So these are the comparison of mass, spin, and magnetic momentum of pions. So from this equation 54, we can say m pi is having an amount of mass, mass that is 270 of m of electron, right? The comparison of masses, spins, and magnetic moments for pions. These are the comparisons for 5 plus. It is 273.2 ME. For pi naught, it is 264.4 ME, and for pi minus, it is same as that of pi plus. Okay. So R is nearly equals to h cross by 2 into 270 m naught c. So it is equals to nearly 1 Fermi. Okay, the range it will go for is 1 Fermi. This is about meson theory of nuclear forces. We will discuss some techniques. So just of phase contrast microscope. First, this is the fourth unit, I think, of your syllabus. Fifth unit, I think, of your syllabus. This is phase contrast microscope. We will try to explain some techniques that is about microscopes, that is electron microscope, phase contrast microscope, and scanning electron microscope, and also TEM, that is transmission electron microscope. We'll discuss those topics today. Let us see what is phase contrast microscope. So the phase contrast microscope is a light microscope. What it is a light microscope. It is It is modification of compound microscope. It is what? It is a modification of compound microscope. Compound microscope is modified and it is made to a phase contrast microscope. Okay. It contains all the components of a compound microscope in addition to an angular ring and a faceplate. So there will be an angular ring and there will be a faceplate. Faceplate will be uh, placed in addition to the compound microscope. Okay. It magnifies not only object but also changes its brightness. So it magnifies the object and also it can bright uh, the sample. Okay, the image. So this is the schematic diagram, that is image plane, diffracted lines, and direct light which is from the source, that is objective. This is specimen. This is condenser. Okay, so there will be a condenser. This is the instrumentation. This phase contrast microscope. So what is the principle behind it? The phase contrast microscope separates the illuminating background light and the specimen scattering light. So what it does, it separates the illuminating background light and specimen scattering light. Phase contrast microscope is used to visualize transparent, colorless, unstained living biological specimens. So it is why we will use this phase contrast microscope, because it uses a colorless, unstained living biological specimens. To visualize these biological specimens, we use phase contrast microscope. Okay, these objects are called phase objects. These are objects are called what? These are phase objects. Light is diffracted and retarded dif, ref, based on the refractive index of the object. Based on the refractive index of the object, highly refractive structures bend and retard light much. Okay, but where there is highly refractive structures, there will bending and retarding of light will be more. Okay, this principle is used in phase contrast microscope. This principle is used in where? It is used in phase contrast microscope. So what is the principle? It separates the illuminating background light and specimen scattering light. Specimen scattering light. And it is based on the refractive index of the object. So the light will be bended and retarded based on the refractive index of the object. There will be high refractive structures will be there. Where there is high refractive structures, there will be retardation and bending of light will be more. Okay. So this is the phase contrast microscope optical train. This is the lamp. This is the collector lens. Through this collector lens, the light will flow, and this is the apertures. These are the two apertures here. So this is the condenser annulus, and this is another condenser, and this is the specimen. This is the objective lens. This is the phase plate, which is diffracted through this phase plate, and it is surrounded by the yellow light, and it is given the image. Okay. Phase contrast microscope is the first microscope method which allow the observation of living cells. So this phase contrast microscopes uh, allowed to see the observation of living cell. Okay, it is invented by Fritz Zerknecht and was awarded Nobel Prize in 1953. Types of phase contrast microscope. There are two types of phase contrast microscopes. They are positive phase contrast, that is dark contrast, negative phase contrast, that is light contrast. Positive phase contrast or dark contrast. In positive phase contrast, the specimen is dark and the background is bright. For the dark contrast, the background will be bright and the specimen will be dark. Okay, the specimen is surrounded by a bright halo. Okay, the specimen will be surrounded by the bright halo in the positive phase contrast. In the negative phase contrast, this will be reversed, I think. In negative phase contrast, the specimen is bright. Here the specimen is dark. Here the specimen is bright, and the background is dark. Here the background will be dark. For positive phase contrast, the background will be bright. The specimen is surrounded by a dark halo. Here the specimen is surrounded by bright halo. Here the specimen is surrounded by dark halo. Okay. This is the structure. Phase contrast microscope has the following parts. This is light source, that is tungsten lamp. So and annular diaphragm, and condenser, and objective lens with faceplate, and body tube, eyepiece, body tube, and course adjustment. There will be adjustment for this one. Fine adjustment, stage, and nose piece, arm, inclination, and base. So this is the base. These are the base lens. These are the filter holder which filters. This is the condenser here. This is mechanical stage where we keep the where we can adjust. These are the objective lens. This you can observe here. These are the objective lens. This is binocular heads. Through these lenses we can see the visual of this sample actually. So these are the eyepieces here. This is the C mount. Binocular head. This is lock screw. We can adjust and we can lock that adjustments here. Arm stand, force focusing knob, fine focusing knob here. The mechanical stage control here. This is the mechanical stage control. Light dimmer, where you can dim the light, and this is on and off switch. This is the base. Okay. This phase contrast microscope. Annular diaphragm. If you see the annular diaphragm, annular diaphragm is a plate having circular thin area in the center surrounded by thick areas. So this is a having circular thin area in the center surrounded by thick areas. Okay, this is condenser annular. Here, if you see, this is the circular thin area in this center surrounded by thick areas. Hence, light coming from the source can pass through diaphragm in the form of a circle. So the light passes through this diaphragm will be in the form of circles. The portion coated with light retarding material, portion devoid of light retarding material. These are the two things you can observe here. The annular phase plate is a transparent disc with a groove. Okay, the size of the groove is identical to the circular area of the substage diaphragm. The annular groove of faceplate filters of 70 to 90%, waves, S waves, and leaves D waves as such. Okay. It is placed at the back focal plane of the objective. Condenser. If you go for the condenser, this is the condenser adjustment, alignment of the condenser along the microscope optical axis. It gathers and focuses the reflected light on the object. The condenser has a diaphragm. This condenser will be having a diaphragm again. It allows the required intensity, power, concentration, strength of light pass through it. So it allows the required intensity, how much intensity it is needed, we can allow through this condenser. Okay. Diaphragm is a 5-block disc placed under the stage. Each hole is of a different diameter. By turning it, you can vary the amount of light passing through the stage opening. How much of light want to pass through that, we can pass through this diaphragm. Nose piece and aperture. Nose piece is a rotating. Here, this is the nose piece, and here it is an aperture. Here, nose piece is rotating turret that holds the objective lenses. The viewer spins the nose piece to select different objective lens. The aperture is the middle of the stage that allows the light from the illuminator to reach the specimen. Through this aperture, it will reach to the specimen, and it falls and it will give the radius. Objective lens. Metal cylindrical cylinders attached below the nose piece contains special ground and polished lenses. It magnifies the object. So these objective lens will magnify the specimen, and we can see the nucleus and this very small things here. Cell wall, cell membrane, vacuole. Types of objective lens. There are types. There will be types of objective lens, that is low power objective, high power objective, oil immersion objective. Low power objective lenses gives the lowest magnification, usually up 10x. High power objective is gives higher magnification, usually from per 40x to 43x. Oil immersion objective gives the highest magnification. It gives the highest magnification comparatively with the high power and low power objective lenses. This is 97x to or 100x and is used cedar wood oil or synthetic oil for the better resolution. Okay. It magnifies the image produced by the objective lens. Eyepiece, what it will do, it magnifies the image produced by the image produced by the objective lens will be magnified using this eyepiece. Okay. If you see, this one is a virtual actually. So this is subject. So this is the retina. From there, simple magnifying lens will be there. Actually, this is a subject, but we can see a virtual image like this. Body tube. It is a tube with objective lens at lower end and the eyepiece lens at the upper end. So in the lower end there will be objective lens. In the upper end there will be eyepiece. So this is the bridge between this body tube will be the bridge between this objective lens and the eyepiece. Okay. Force adjustment. It moves the body tube up and down rapidly to correct the distance from the object to get focused. Okay. Fine adjustment. So these are the fine adjustment and force adjustments are there. It moves the body tube up and down slowly to make the exact focusing. For the focusing, there will be fine adjustment. To move the body up and down, the coarse adjustment is needed. Okay. Specimen stage. It is a platform with a hole in the center. So it is having a hole in the center. The light falls on the object through the holes. The slide is placed on the stage. Okay. The slide will be placed on the stage. This is the sample holder. This is the stage. So the slide will be placed on that stage. This on the stage. These are the graduated locator markings. X-ray translation control knobs. This is the stage opening, stage lock control, stage clips. The stage clips holds the slide firmly on the stage. So this is the nose piece. It is the form of rotating this, having holes for the fitting of the objective lens. For the fitting of the objective lens, we will use nose piece. Inclination joints. It permits tittling of upper part of the microscope to adjust the eye level. To adjust the eye level, inclination joint is needed. So you can see here it is eyepiece. This is the body tube which is connecting objective lens and eyepiece. And this is the coarse adjustments of the knob. These are the fine adjustments of the knob, adjustment knobs. These are the objective lens here. This is the stage. This is the stage. And these are the condensers. This is the substage. And this is the mirror. And this is the base. The inclination joint is there. Revolving nose piece is there. Base or foot. The foot keeps the body in position. The foot keeps the body in the position. Okay. Use of phase contrast microscope. What are the uses of this phase contrast microscope? It is used to be the cellular parts of the protozoa, bacteria, and others without staining. Okay. So it can observe protozoa, bacteria, cellular parts of protozoa, bacteria. Okay. Employed to observe cells in the culture without fixation and staining. Okay. Used to examine water and other fluids for the presence of minute transparent microbes, bacteria and flagellates. So this is used to observe water and other fluids for the minute transparent microbes. Okay, that is bacteria and flagellates. Used to study phagocytosis, pinocytosis, cell divisions, etc. in cells. used to study behavior of chromosome during mitosis and meiosis. Okay, this is about the phase contrast microscope. Next, we will go for the electron microscope. Next, we will go for the electron microscope and scanning electron microscope. Let us see. So, what is my electron microscope actually? So, microscope that uses a beam of accelerated electrons as a source of illumination, high resolution of images, able to magnify objects in a nano. So actually this electron microscope uses a beam of accelerated electrons, okay, as a source of illumination. So the electrons will be used to detect the, uh, sorry, to know the high resolution images of, to get the high resolution images of any specimen or anything which we need, okay, in a nanometer ranges, okay. So it was discovered by uh, Ernest Ruska in 1906 to 9. He was born in 1906 and died on 1988. A German engineer and academic professor built the first electron microscope in 1931. When it was discovered on 1931 by Ernest Ruska. The same principles behind this prototype still governs modern EMs. Okay, electron microscopes. The same principle which was used by the Ruska in 1931, those principles only used in the modern electron microscopes also. Okay, this is the electron microscope. You can observe. This is the electron microscope. You can observe here. So we will see. So what are the parts of this electron microscope? Form of a tall vacuum column that is vertically mounted. Okay, electron gun. First part is electron gun. The electron gun is a heated tungsten filament which generates electrons. So electron gun is what? Electron gun is a heated tungsten filament. Electron gun is a heated tungsten filament which generates electrons. From this electron gun only the electrons will be generated and it is fall on that sample or specimen what uh, we use, okay, to find the uh, structures, okay. Electromagnetic lenses. Second part is electromagnetic lenses. The condenser lens focus the electron beam on the specimen. So the condenser lens, the electron gun will eject the electrons onto the condenser lens, and this condenser lens will be focused to fall on the specimen. Okay. A second condenser lens forms the electrons into a thin, tight beam. So there will be another condenser lens which will form the electrons in a thin, light beam. Okay. The electron beam coming out of the specimen passes down the second of magnetic coils called objective lens. So the electron beam from the sample or specimen will again passes down to the second of magnetic coils called objective lens. There will be magnetic coils will be there. Those are called objective lens. From the sample, from the specimen, the electrons will be called on the objective lens. Okay. Through that objective lens, it will be going. Okay, which has high power and forms the intermediate magnified image. So these lens will form an intermediate magnified image. So first the electron gun will eject the electrons onto the specimen. The specimen, sorry, it will pass on from the condenser and focuses onto the specimen. After this condenser lens, again another condenser lens will be there, which will make the electrons thin light beam, and again it will passes through the second magnetic coils called the objective lens through the sample, through the specimen, and these objective lens will give the intermediate magnified image. Okay, the third set of magnetic lens are called projector ocular lenses, produce the final further magnified image. from that objective lens, as the objective lens makes the intermediate magnetic magnified image, the third set of magnetic lens will the projector. What we will do, the projector lenses produces the further magnified image. The This is the intermediate magnified image which is formed by the objective lens. From those lenses, again, these electrons will this image or the electrons will go through these projector lenses and they will form a magnified image. Okay, each of these lenses acts as an image magnifier, all all the while maintaining an incredible level of detail and resolution. Okay, so this makes the incredible level of detail and resolution of that specimen. Parts of electromagnet, as this one is electron gun, these are the electromagnetic lenses, and the third one is a sample holder. The specimen holder or sample holder. Specimen holder is an extremely thin film of carbon or colloidal held by metal group. This is made up of, the sample holder is made up of carbon. Okay, image viewing and recording system. The final image is projected on a fluorescent screen. So the image which has formed by the secondary magnetic lenses or projector lenses will be imaged, projected on the fluorescent screen. There will be a fluorescent screen. On that fluorescent screen, it will be imaged. Below the fluorescent screen is a camera for the recording of that image. There will be a camera below that fluorescent screen that will record the image. Okay. Working principle of electron microscope. The electron gun generate electrons. Two sets of condenser lenses focus on the electron beam on the specimen and then it into a thin tight beam. So after the projection of these electrons from the electron gun, they will be projected through these two condenser lenses. So these condenser lenses will make the electron beams as thin tight beam. Okay, it will make it to thin. Okay, to move electrons down the column, and it is focused onto the specimen actually by these condenser lenses. Those condenser lenses, they will do, the electron beams which are coming out of the electron gun were made to thin tight beam and focused onto the specimen. To move electrons down the column, an accelerating voltage, mostly between 100 kV to 1000 kV, is applied between the tungsten filament and anode. So there will be voltage applied between this tungsten filament and anode. Okay, the specimen to be examined is made extremely thin. The specimen which has to be examined is very thin actually, at least 200 times thinner than those used in the optical microscope. So it is 200 times thinner than the optical microscope. The specimen which has to be examined, it will be 200 times thinner to that used in the optical microscope. Ultra thin sections of 20 to 100 nanometers are cut, which is already placed on the specimen holder. So there will be ultra thin sections of about 20 to 100 nanometers were cut, which will be placed on the specimen holder. Application of these electron microscope. So investigate the ultrastructure of a wide range of biological and inorganic specimen. To investigate the ultrastructure of biological and inorganic specimen. So biological and inorganic specimen structure can be investigated by using this electron microscope. Used for quality control and failure analysis. Can be used for quality control and failure analysis. Modern electron microscopes produce electron micrographs. So using specialized digital cameras and frame grabbers capture the images. So these model electron microscopes were produced, electron micrographs, using specialized digital cameras. Nowadays we are using the digital cameras and frame grabbers capture these images. Okay. Study of microorganisms like bacteria, virus, and other pathogens have made the treatment for these diseases very effective. So Study of microorganisms can be done by using this electron microscope. Advantages of electron microscope: very high magnification. It can do incredible high resolution. Material rarely distorted by preparation. It is possible to investigate a great depths of field. So diverse applications will be there by this electron microscope. What are the limitations? Let us see the limitations or drawbacks of this electron microscope. Leaf specimen. The leaf specimen cannot be observed. The object should be ultra thin. The live specimen cannot be observed here by using this electron microscope. And also the objects must be ultra thin. It must be ultra thin. The specimen is dried and cut into ultra thin sections before observation. As the electron microscope works in a vacuum, the specimen should completely dry. So it must be completely dry, which should be worked in a vacuum. Expensive to build and maintain. Electron microscope is expensive, requiring research training. So to control, to know this, we need some researcher training for the electron microscope. Image artifacts resulting from specimen preparation. This type of microscope is large, cumbersome, extremely sensitive to vibration and external magnetic fields. Okay. Types of electron microscope. There will be two types of electron microscope: transmission electron microscope, scanning electron microscope. So electron microscope uses the actually electrons, okay, high intensity electrons, which can be which are subjected to fall on the specimen or sample, so that we will get the images. So the same principle is used in these two microscopes, that is transient electron microscope, scanning electron. The purpose of these microscopes may be changes, but the principle is mostly same. So transmission electron microscope. Let us see the transmission electron microscope. That is mostly we'll call it as TEM, TEM, transmission electron microscope. Powerful electron microscope that uses a beam of electrons. So it is a powerful electron microscope that uses beam of electrons to focus on a specimen, producing a highly magnified and detailed image of the specimen. So in the TEM, again we will use beam of electrons, which will be focused on the specimen, which are highly magnified, and there will be a detailed image of that specimen. Okay, there will be a detailed image of the specimen. The magnification power is the magnification power is over 2 million times, producing the image of specimen, which enables easy characterization of the image in its morphological features and compositions. So what is the principle actually? The beam of high energy beam of high intensity beam of electrons are focused onto the specimen, and they will produce highly magnified and detailed image of the specimen actually. Okay, so the magnification power of this transient electron microscope is 2 million times. It is the magnification power is over 2 million times of the image actually, of the specimen actually. So it will give 2 million times more magnification to the original one, producing the image of the specimen. Okay, which enables easy characterization, so that the magnification of that sample is more, we can easily characterize the morphological features and structures, and what are the compositions which are in the sample. So we can study easily through this magnification. Okay. So principle of transmission electron microscope. What is the principle actually? So We use electron microscope, electron beams only, but what is the principle behind? In the electron microscope, what happened? It will be there will be electrons which are passed through the specimen, and there will be magnification of light. Magnification of that specimen happens through this through those lenses, right? Magnetic lenses. But here, similar to the light microscope, it will be similar to the light microscope. Microscope uses light rays to focus and produce the image, while the TEM uses a beam of electrons to focus the specimen, produce an image. So light microscopes use light rays. Here the TEM uses a beam of electrons, which will be focused onto the specimen to produce an image. Electrons have shorter wavelength in comparison to light, which has a long wavelength. Light will be having a long wavelength, which will move far, but the electrons are having shorter wavelength. So better resolution will come. So this is the transmission electron microscope. This is the electron source. These are the condenser lenses. These are two condenser lenses. This is the condenser aperture. This is the sample. This is the objective lens, objective aperture, selected area aperture, the intermediate lenses, projective lenses, and fluorescent screen. This is the fluorescent screen where the image of that sample or specimen can be observed. Okay. From this electron source, the light will fall through these condenser lenses onto the condenser aperture onto the sample, and the sample will be the beams which are coming out of this sample will be uh, subjected onto the objective lens, where the objective lens forms an image into the objective aperture, and that selected area of aperture is again formed onto the intermediate lenses. From the intermediate lenses, there will be a projective lenses where it will be magnified multiple times uh, uh, and magnified highly, and it will be projected onto the fluorescent screen. So if you see the applications of transmission electron microscope, then to visualize and study cell structures of bacteria, viruses, and fungi. So we can study the cell structures actually. So structures of what? Bacteria, viruses, and fungi. So by using this transmission electron microscope, we can visualize and study the cell structures of bacteria, viruses, and fungi. To view bacteria, flagella, and plasmids, we can see the bacteria, flagella, and plasmids. Okay. To view the shapes and size of microbial cell organelles. So we can see the shapes and sizes of microbial cells and organelles. To study and differentiate between plant and animal cells, we can differentiate the plant and animal cells also. It is also used in nanotechnology to understand nanoparticles such as zinc oxide nanoparticles. So by using this transmission electron microscope, we can study the nanoparticles such as zinc oxide nanoparticles, which is used to detect and identify fractures, damage, microparticles, which further enable repair mechanism of the particles. So these are also useful to identify the fractures, damages which are happened in the microparticles. So which can detect the particles damages, and uh, and it can repair. Okay, they can repair by identifying these fractures. Advantages of transmission electron microscope. It has a very powerful magnification of about 2 million times that of the light microscope. So light microscope, comparatively to the light microscope, the transmission electron microscope magnifies the specimen 2 million times. Okay, it can be used for a variety of application ranging from basic biology to nanotechnology to education and industrial uses. So it can be used for a variety of applications. Okay, from basic biology to nanotechnology, to education and industrial uses. We can use in it. This term is used in industrial areas also. It can be used. It can be used to acquire vast information on compounds and their structures. For finding the structures of the compounds, we can use this term, transition electron microscope. It produces very efficient, high quality images with high clarity. So produces high quality images, okay, with high clarity. By using transmission electron microscope, we can see high quality images and high clarity images of the specimen. It can produce permanent images. It can produce permanent images. It is easy to train and use the transmission electron microscope. So it can be easy for the electron microscope. It is difficult, but for the transmission electron microscope, it is easy to train and use the transmission electron microscope. Limitation of transition electron microscope. So actually, these PEMs are very expensive. This is one of the drawbacks. PEMs are very expensive to purchase. They are very big to handle. So transition electron microscopes are very big to handle. The preparation of specimens to be viewed under the transmission electron microscope is a very tedious. So the use of chemical fixations, dehydrators, the embedments can cause the dangers of artifacts. They are laborious to maintain. It requires a constant inflow of voltage to operate. It means constant inflow of voltage. Okay. They are extremely sensitive to vibrations and electromagnetic movements. Hence, they are used in isolated areas where they are not exposed. So, they are used in isolated areas only. So, these are extremely sensitive actually. These transition electron microscopes are extremely sensitive to vibrations and electromagnetic movements. So, it cannot be exposed. These cannot be exposed to uh, in an exposed area. These must be in isolated areas. It produces monochromatic images unless they are fluorescent, unless they use a fluorescent screen at the end of the visualization. If you use a fluorescent screen at the end of visualization, it cannot. Uh, it it will produce if only monochromatic images if we not keep the fluorescent screen at the end of the visualization. These are the transmission electron microscope images actually. Scanning electron microscope. This is another type of electron microscope. So type type of electron microscope that scans surface of microorganisms, that uses a beam of electrons moving at low energy to focus and scan specimen. Here it is a type of electron microscope that scans the surface of microorganisms. So this electron microscopes scans the surface of microorganisms that uses a beam of electrons. So it will use a beam of electrons moving at low energy to focus and scan specimens. So these also use electron beams only, but the electrons moving at low energy focus the and scan the specimen. Principle and scanning electron microscope. Principle of scanning electron microscope. Let us see. Applying kinetic energy to produce signals on the interaction of the electrons. So we will apply the kinetic energy to produce signals on the interaction of these electrons. These electrons are secondary backscattered and diffracted backscattered electrons, which are used to view crystallized elements, and photons. So there will be three type of beams which will be coming out of the sample actually. Those are So we will apply actually firstly we will applying kinetic energy to produce signals on the interaction of the electrons. Okay, we will apply the kinetic energy which can produce signals where there is interaction of these electrons to the specimen. These electrons are secondary backscattered. So these are the secondary backscattered and diffracted backscattered electrons which are used to view crystallized elements. Okay, so diffracted backscattered electrons are there, which are used to view crystallized elements and photons. One is secondary backscattered, one is diffracted backscattered, and other secondary and backscattered electrons is used to produce an image. Okay, these secondary and backscattered electrons, which are coming from the specimen, are used to produce an image. The secondary electrons are emitted from the specimen. The secondary electrons will be emitted from the specimen, play the primary role of detecting the morphology and topography of the specimen, while the backscattered electrons show contrast in the composition of the elements of the specimen. So the secondary electrons which are coming from the specimen will detect the morphology and topography of the specimen. So those will detect the morphology and topography of the specimen, while the backscattered electrons, secondary backscattered the diffracted backscattered electrons show the contrast in the composition of the elements of the specimen. So, morphology and topography are can be detected by secondary electrons actually, and the composition of these elements, these elements can be studied by using these backscattered electrons. Okay, so actually, when the beam of light falls on the specimen, there will be backscattered electrons and secondary electrons. These secondary electrons, what will do? It will give the morphology and topography, whereas the backscattered electrons will give the composition of those elements which are forming the specimen. This is scanning electron microscope. This is the electron source, condenser lens, aperture, scanning coil, objective lens, backscattered electron detector, and the SNAP. This is the detector which is connected to the system amplifier and to the reading system. Okay, the computer. This scan generator. So the light coming from the electron source will go through this condenser lenses and to the aperture and through the scanning coil and it will give to the objective lens which will form an image. Sorry, which will be given to the objective lens, and backscattered detector will be there, and it will go onto the specimen and the electrons will be ejected from the specimen, backscattered and secondary electrons. Secondary electrons will give the morphology and topography, whereas the backscattered electron will give the composition of those elements. This is scanning electron microscope. You can observe. So application of scanning electron microscope. It is used in a variety of fields, including industrial uses, nanoscience studies, biomedical studies, microbiology. So these scanning electron microscopes are used in variety of fields. So it can be used in industries, nanoscience studies, biomedical studies, and microbiology. Used for spot chemical analysis in energy. So dispersive X-ray spectroscopy. So it can be used in chemical analysis in energy, dispersive X-ray spectroscopy. Used in the analysis of cosmetic components, which are very tiny in size. So these are used in the cosmetic components also, which are used in a very tiny size. Used to study the filament structures of microorganisms. They are used to study the filament structures of microorganisms. Used to study the topography of elements used in industries. So This can be used. This scanning electron microscope can be used to study the topography of elements which are used in the industry. Advantages of scanning electron microscope. They are easy to operate and have user-friendly interfaces. So these are easy. This can be operated easy, and these are having user-friendly interfaces. They are used in a variety of industrial applications to analyze structures. Sorry, they are used in variety of industrial applications to analyze surfaces on solid objects. Okay, so to analyze the applications of to analyze the surface of solid objects, these can be used. Scanning electron microscopes can be used. Okay, some modern scanning electron microscopes are able to generate digital data that can be portable. So there are some modern SIMs which can port the data, digital data. Which can generate digital data can be ported. Yeah. It is used to acquire data from the SIM within a short period of time of about 5 minutes. So it can acquire the data within a short period. That is up to 5 minutes. It can give the data of that sample or specimen. Limitations. Let us see the limitations. They are very expensive to purchase. So these are very expensive to purchase, as the TEM is also a very expensive electron microscope. This one is also a very expensive one to purchase. They are bulky to carry. They are very bulky to carry. So they must be used in rooms that are free of vibrations and free of electromagnetic elements. Same as the transition electron microscope, these are also uh, used. These must be also used in the rooms which are free from these vibrations of electromagnetic elements. Okay, they must be maintained with a consistent voltage. These must be maintained at a consistent voltage. The fluctuations may defect the instrument. They should be maintained with access to cooling systems. The cooling systems must be needed for the maintenance of this scanning electron microscope. These are the images from the scanning electron microscope. So let us see the differences between the scanning electron microscope and transmission electron microscope. So scanning electron microscope, the scanning electron microscope provides the information on sample surface and its composition. So what is the use of this transmission electron microscope then? SEM provides the sample surface and composition, whereas this TEM are used to study the structure, morphology, and stress. Okay, SEMs use a specific set of coils to scan the beam in a raster-like pattern and collect the scattered electrons. So SEM are used for the sample surface to provide the sample surface information and composition, and these SEMs will be used a specific set of coils. So to scan the beam in a raster-like pattern, okay, and collect the scattered electron. By collecting the scattered electron, we can find the surface and composition. Okay, in the TEM, we use transmitted electrons. In the same, in the scanning electron microscope, what happens? In the scanning electron microscope, we will see the scattered electrons from the specimen. Actually, we will see the scattered electrons from the from the specimen, and we will try to study these scattered electrons, and we can by this study of the scattered electrons, or this can give the surface uh, sample surface and composition. Okay, but here the TEM is transmitted through the specimen. So use the transmitted electrons. The electrons which are passing through the sample before they are collected. So these are which are passed through the sample before they are collected. So TEM offers invaluable information on the inner structure of the sample. It gives the inner structure of the sample. This TEM will give the inner structure of the sample, but the SEM will give the only sample surface and its composition. Inner surface structure of the sample, such as crystal structure, morphology, stress state information. Okay. Scanning electron microscope is gives the resolution limited to 0.5 nanometers. SEM resolution is limited to 0.5 nanometer. Then transition electron microscope has a resolution of even less than 50 pm picometer. So it can give the resolution up to 50 picometers. Okay. So if you want to get the information on the surface of your sample, like roughness or contamination detection, then you should choose a SEM. So for the information on the surface of the sample, if you want any information, you can use SEM, like roughness, contamination detection. We can use the SEM here. If you would like to know what the crystal structure of your sample is, or if you want to look for possible structural defects or impurities, then using a TEM is the only way to do so. So if you want some structural defects, if you want to find some possible structural defects and impurities, if you want to study the internal structure of any sample, you can use this TEM, transmission electron microscope, because transmission electron microscope ejects the electrons and the electrons will be transmitted through the sample. But here in the SEM, scanning electron microscope, the electrons will be scattered. Through that scattered electrons, we'll try to study the structure or sample surface in composition. SEMs provide a 3D image of the surface of the sample. So it will give the 3D image of the surface. SEM images are 2D projections of the sample, which in some cases make the interpretation of the results more difficult for the operator. So SEM images will be 2D projections. SEM imaging, there is no specific requirement of the sample preparation. Scanning electron microscope imaging, there is no specific requirement of the sample preparation. Okay. Due to the requirement of transmitted electrons, TEM samples must be very thin. So for the SEM imaging, there is no specific requirement, but for the transmission electron microscope, the sample must be very thin, generally below 150 nanometer. The samples must be about below that of 150 nanometers. Okay, size must. And in case that high resolution imaging is required, even below 30 nanometers. So there will be need of high imaging is required below 30 nanometers. Scanning electron microscope samples require little or no effort for sample preparation. So for the sample preparation of scanning electron microscope, there will be little or no effort for the sample preparation. It can be detected imaging by mounting them on the aluminum stub. So it can be detected when it is mounted on the aluminum stub. TEM sample preparation is quite complex and tedious procedure that only trained and experienced users can follow successfully. The samples need to be very thin, as flat as possible. So the sample must be very thin and very flat. Okay. But here it is not, there is no need of the preparation here. Mostly little or no more need of the preparation of the sample. But here for the TEM, the sample must be very thin and it must be flat as possible. And preparation technique should not induce any artifacts such as precipitates and amorphization to the sample. So there will not be any induce of any artifacts like this precipitations and amorphization to the sample. Many methods have being developed including electro-polishing, mechanical polishing, focused ion beam milling, dedicated grids and holders are used to mount the TEM samples. So these are the techniques which are used to prepare the samples. Okay, these are the techniques which are used to prepare the samples. SEMS usually use accelerated voltages up to 30 kilovolts. Okay, SEMS usually use accelerations voltages up to 30 kV. TEM uses in the range of 60 to 300 kV. The magnifications of the for the SEM is limited. The magnification for the SEM is limited up to 1 to the magnifications for the SEM is limited up to 1 to 2 million times. The magnifications that TEMS, that is transition electron microscopes, is much higher compared to SEMS. TEM users can magnify their sample by more than 50 million times. So it can do the magnification 50 million times. However, the maximum field of view, that is maximum field of view that SEMS can achieve is far larger than TEMS. Okay. Similarly, the depth of the SEM systems are much higher than the TEM systems. Users can only use image a very small part of their sample. So we can use only small part of their sample to image, to view. Okay. But here there will be maximum field of view for the SEMS. In addition, the way images are created are different in two systems. In SEMS, samples are positioned at the bottom of the electron column and scattered electrons. In SEMS, samples are positioned at the bottom of the electron column and scattered electrons. They are captured by electron detectors. Photo voltage signal which is amplified and gives rise to image on a PC screen. Okay. There will be different in two systems. Different in two systems. The same samples are positioned at the bottom of the electron column. Okay. Then the scattered electrons are captured by the electron detectors. Electron detector means captured the scattered electrons. So these scattered electrons are used by these photomultipliers convert into this voltage signals. Okay, which can be amplified and which can be imaged on the PC screen. Transmission electron microscope. In a transmission electron microscope, the sample is located in the middle of the column. So the sample will be located in the middle of the column. The transmitted electrons pass through and through a series of lenses below the sample. So below the sample, there will be some lenses like intermediate and projector lenses. An image is directly shown on the fluorescent screen by a charge coupled device. Camera onto the PC screen. So here the electrons were detected by the electron detectors from the scattering electrons, but here there will be the sample will be in the middle of the instrument, and there will be some lenses, intermediate and projector lenses. Through those lenses, we can form the image, and it can be shown on the fluorescent screen. Okay, there will be cameras available. Relatively simple to operate. The scanning electron microscope is simple to operate. Generally, TEMs are more complex to operate. TEM users require intensive training before being able to operate them. So there will be a need of the training to operate these TEMs. Special procedures. Special procedures need to be performed before every use. This with several steps, including that ensure that the electron beam is perfectly aligned. Relatively simple. This is relatively simple. TEMs may enable much more resolving power and versatility to the user, but they are much more expensive and larger than SEMS and require more effort in order to acquire the interpret results. Okay, so these are uh, They need much more resolving power and versatility to the users. These are more expensive, these stems, but these are scanning electron microscopes are very simple for the resolving all these things, and it is there will be much training is needed for the transmission electron microscope, but here the scanning electron microscopes can be operated easily. Okay, these are the techniques or instruments which are used to study the mm, sample areas of the structure and morphology, and also the internal structure of this any sample. By using these microscopes, we can find out these characteristics. Okay, this is first one is electron microscope. Electron microscope are divided as two. One is SEM and SEM. These use the same principle of that electron microscope, but here we will study the can be used from by the electron to that sample or specimen onto the objective lens, and they will form the image. Here the scanning electron microscope. In the scanning electron microscope, we will study about the scattered electrons. By the scattered electrons, we will study the morphology and topography of the electrons. Okay. So this is about today's class. So we'll meet again with another class. Thank you. Thank you for participating.